The purpose of this video is to explain the applications and skills for topic 3.2 Chromosomes for the IB Biology exams beginning in 2016. In this video, we will cover the applications of comparing the genome size in 5 different organisms or things, the application for comparing the diploid number of chromosomes in 5 species, and the skill of identifying the locus of a human gene and its polypeptide. We will first compare the genome length between a virus, a gut bacteria, a fruit fly, a human, and a woodland plant. To organize the differences, create a data table like this. As a quick review, a genome is the whole of genetic information of an organism. In plants and animals, this includes the whole of all chromosomes and the DNA inside the mitochondria and plastids. For prokaryotes, this includes the naked DNA and any plasmids. Note that plastids and plasmids are not the same thing. The smallest of the 5 genomes we are comparing belongs to the T2 bacteriophage, which is a virus that infects E. coli. It should be noted that this is not technically a living thing because it does not fulfill all characteristics of living things. Fun fact, and something HL students should understand in more detail, this virus strain was used in an important experiment that led to the discovery that DNA, and not protein, was the true genetic material in 1952. It has a genome size of 0.18 million base pairs, or 180,000 base pairs, which is the shortest of all the things we are comparing today. The next shortest genome belongs to the sole prokaryote in our list, E. coli, which is a bacteria that resides in the guts or large intestines of animals and can help synthesize vitamin K and regulate the colonization of other bacteria in the intestines. It is a larger genome than the T2 bacteriophage at 5 million base pairs, making it the smallest genome of all organisms in the list. Now for our eukaryotes. Next on our list is Drosophila melanogaster, a fruit fly, and it has a genome size of 140 million base pairs. Humans, or Homo sapiens, have a genome size of 3,000 million base pairs. Lastly, Paris japonica, a woodland plant from Japan, has the largest genome yet assayed or tested to determine its characteristics. It comes in at a size of 150,000 million or 150 billion base pairs, 19 billion more than the longest size known. In eukaryotes, genome size varies based on the size and number of chromosomes, correlated but not directly proportional to the complexity of an organism. The proportion of DNA that contains coding regions of genes is variable, and the amount of gene duplication varies. That is, that some organisms can contain varying numbers of alleles for the same gene. We will now compare the number of chromosomes in diploid cells of 5 different eukaryotic species: a worm, rice, a human, a chimpanzee, and a dog. To organize the differences, create a data table like this. As a quick review, the term diploid refers to having 2 chromosomes of each type, which is the result of fertilization between 2 haploid cells. Another way to describe this is the cell contains a pair of homologous chromosomes of each type. All eukaryotes have at least 2 different types of chromosomes, making the diploid number at least 4. This is the case for the horse threadworm, Familiaris equorum, which has a total of 4 chromosomes or 2 pairs. Rice, or Oryza sativa, has a remarkable approximately 41,000 genes organized on only 12 pairs or 24 chromosomes. Compare this with humans, Homo sapiens, which have only 23,000 genes organized on 23 pairs of, or 46 chromosomes. Our not too distant relative, pan troglodytes or chimpanzee has 24 pairs of or 48 chromosomes. Lastly, dogs, Canis familiaris, have 78 chromosomes. With each of these species, the sizes of each chromosome vary from large to small, and we will make observations and generalizations based on the sizes and numbers of chromosomes once we look at karyograms. Once you have your table completed, I want you to look up why the diploid number of chromosomes follow this curious pattern of all being even numbers. Why is the diploid number of chromosomes an even number and not an odd number? Last but not least, I want you to find the locus of a human gene on its specific chromosome. For this, you will use the same gene that you used to compare the sequence between different species. For example, I demonstrated the assignment using HBB, or one of the gene that produces part of the hemoglobin polypeptide in red blood cells. You will need to go to omim.org to complete this. Read the description in the purple box on page 154 of the ebook. When you get the gene map, you will be presented with a lot of information. I want you to record the following information down: the location, the number of BP or base pairs, and a description of the gene's function. In order to explain the location, scientists follow a common convention. To demonstrate this, I will use the gene HBB. It has a locus of 11p15. What this means is the following. 11 refers to the human chromosome the gene is located on. In this case, chromosome 11. P refers to which arm of the chromosome the gene is located on. In this case, it is located on the P arm or short arm. Q refers to the long arm of the chromosome. 15 refers to the specific location of that gene on the arm. The greater the number is, the further away from the centromere the gene is located. In your assignment submission, I want you to include a screenshot of the window with the relevant information. It should look like this.